Hi everyone, this is lecture 23, Aneuploidy, and the material here is found in chapter 16 in your textbook, page 476 to 483. We've talked a lot about cell division lately, mitosis and meiosis. Let's see what happens when those processes go wrong. It's always fun to talk about what happens when stuff goes wrong, right? A few questions to begin. Number one hypothesis about the correlation between a mom's age and the increased frequency of Down syndrome is that. Number two, the number of bar bodies that would be detected in an individual with an XXY chromosome set is how many? And number three, for a species with a diploid number of 18, how many chromosomes will be present in the nuclei of individuals who are trisomic? First, I'm going to introduce a topic called non-disjunction. It's a terrible word. Unfortunately, with the genetics, we occasionally encounter words that are just terrible names. However, it's understandable if I take a minute to explain it. Non-disjunction simply refers to the gain or loss of a chromosome, and this process happens during meiosis. This is a spontaneous type of mutation, and this can happen, for example, in this case in meiosis 1, in the first cell division, and it typically happens when the meiotic spindle fails. It fails to bind to one of the chromosomes. There can also be problems with recombination between the homologous chromosomes. That can also lead to non-disjunction. So what does that word mean? Well, when chromosomes come together in meiosis, in metaphase, it's called joining. When they separate in anaphase, it's called disjoining. And when they fail to separate properly in anaphase, we use the word non-disjoining or non-disjunction. Like I said, it's a terrible word, two negatives, but this picture may help to illustrate it. Here is a normal cell at the top here, which is going to go through meiosis. This would be a precursor cell to a germ cell, for example, an egg or a sperm. During the first cell division, the homologous pairs will come together in metaphase and separate in anaphase. However, if the meiotic spindle breaks down or recombination fails to happen, the two homologous chromosomes may end up going into the same daughter cell, leaving a second daughter cell without one of those homologous chromosomes. This is the failure of anaphase, the failure of separating the chromosomes, the failure of segregation. And so we call this non-disjunction. Metaphase, or meiosis 2 then proceeds, leading to a gamete that has two copies instead of one, or no copies. This process could also happen in meiosis 2, where you have a perfectly normal cell division in meiosis 1, but then the sister chromatids fail to segregate into two daughter gametes. Instead, both of the chromatids go into one of the two gametes. When these gametes that are do not contain the proper complement of chromosomes, when these gametes fertilize another gamete, which is normal, a normal haploid gamete, you end up with zygotes that have the improper number of chromosomes. In some cases, there may be 3 of the chromosomes. In other cases, there may be only 1. When a zygote has 3 of one particular chromosome type, we call that a trisomic zygote or a trisomic individual. The other case here is monosomic, where 1 chromosome is present and when there should be 2. So, fertilization results in an aneuploid zygote. We use this word aneuploid to refer to the improper number of chromosomes. one extra or one less. In contrast, we use the word euploidy to refer to the correct complement of chromosomes. So generally speaking, aneuploidy is the gain or loss of a single chromosome, but not the complete set. When we had introduced the word or the formula 2 times n for diploid cells, we refer to monosomic individuals, those with monosomies, as 2n minus 1, the loss of one chromosome. Trisomic individuals have gained a chromosome, so their genome complement is 2n plus 1. Well, as you might guess, most examples of aneuploidy are lethal, meaning they lead to embryonic death. So the babies typically are never born. The organism is, the individual is never properly developed. Lethality usually is caused by genes not being expressed to the right level, or maybe too much in the case of trisomy. Unbalanced gene expression causes big problems in early development. With humans, autosomes, meaning chromosomes 1 through 22, may have trisomies, but never have monosomies. All monosomies are lethal. And again, that's because of the proper dosage of gene expression is not achieved, and lethal alleles are unmasked a lot of the time. So losing entire chromosome is a bad thing for humans. However, in trisomies, there are a few trisomies in humans that persist to develop but with problems. We'll talk about some examples. Now, sex chromosomes, on the other hand, the X and the Y, tend to have more aneuploidies, and they are less harmful in humans. And the reason why is because humans are set up to already inactivate one X chromosome. So all females have two X's, but only one is active in the adult. And there's a small Y chromosome that doesn't have a lot of genes on it. So losing a Y chromosome is not the end of the world. Let's look at some examples. Here's an example of a monoploidy, sex chromosome monoploidy in humans called Turner syndrome. The karyotype is called 45,X because there is one missing X chromosome. And you can see that in this karyotype here. There's only one X chromosome and the other one is lost. This individual would be female because there is no Y chromosome. But however, development is typically affected. Even though normal females will have only one X chromosome expressed, Turner syndrome does cause problems, in part because early development requires the presence of two X chromosomes. X inactivation happens on about the 12th day of human development. Prior to that, female humans require two X chromosomes. In addition to monosomies, trisomies can happen with the X chromosome. For example, this syndrome called trisomy X. The karyotype is 47,XXX. Again, female because there is no Y chromosome. And variable developmental problems, or some people might be perfectly normal and may never know that they are trisomic. 47,XXY is also found, and that's called Klinefelter syndrome. Because this individual has a Y chromosome, this makes them a male. But development is affected because of the presence of the extra X chromosome. And lastly, the 47,XYY condition is also seen in humans. This is called Jacob syndrome. With the presence of the extra Y chromosome, some individuals may be tall because of extra testosterone, may be aggressive, and may exhibit antisocial behavior. Sorry, guys. That is not universally true, though, and many people with XYY will never know they have it and will be perfectly fine. It's interesting when you compare the normal population, which is this control population in this table, that the frequency of the XYY condition is about 0.1%. However, when you select for people who are in either mental institutions or prisons, the frequency goes up to about 2%, and shown in the second row right here. So whereas people in these facilities 98% of the time will have a normal genotype, about 2% of the time you will find the XYY. When you restrict for height, meaning people who are over 6 feet tall, then that number shoots up to about 4.5%, 4.61%. So people who are tall and in prison or in mental facilities are more likely to have the XYY, even though it is relatively a small percentage of the overall population. Again, sorry guys. So what's really interesting about the sex chromosome aneuploidies is that humans have the X inactivation to help adapt to that extra X chromosome. Here on the top left is a cell that has no bar bodies, and normal males with the chromosome XY and Turner syndrome females with 1X have no bar bodies. We use this formula n-1, where n is equal to the number of X chromosomes. Females, 46XX, will have one bar body, because one of those bar bodies is an inactivated X chromosome. However, interestingly, males have the ability to make bar bodies, as evidenced by Klinefelter's individuals, XXY. That extra X chromosome is inactivated at the 12th day of development. Some people can have 2 bar bodies or 3, depending on how many X chromosomes they have. For autosomes, there are a few examples in humans where trisomies are tolerated, and the best known example is Down syndrome. The karyotype is 47,21 plus, because chromosome 21 occurs in 3 copies, not the normal 2. This is a karyotype for a Down syndrome person. Typically has mental retardation. Other problems include heart defects, early aging. It's relatively high incidence, and it's tolerated in part because chromosome 21, as you can see here, is very small. A small number of genes, relatively speaking, are affected. Down syndrome, along with other trisomies, occurs as a function of maternal, or increases in frequency as a function of maternal age. For moms who are 35 years of age, the incidence of having a child with Down syndrome is 3 out of 1,000. This number shoots up in 5 years at age 40 to 10 out of 1,000, 5 years later to 1 over 30, and 5 years later after that, 1 out of 15 births. Why is this? Well, it's interesting to note that 95% of all Down syndrome cases result from an extra maternal chromosome. So the non-disjunction occurred in the mom. And the reason for this is that the meiotic spindle breaks down over time. Oocyte formation in females occurs in the embryo. These precursor egg cells are arrested at meiosis 1. The meiotic spindle is formed and holds the chromosome in this meiosis 1 stage. Then the eggs finish development at ovulation. Those eggs then may have lost their meiotic spindle at that point, at which point then the aneuploidy would have occurred. So the longer the mom waits to have children, the more likely chance that the spindle will have broken down, increasing the risk for non-disjunction and then aneuploidy. Okay, that's all. I will see you in class.